Hello and welcome to the GCSE Biology tutorial. This is for topic B6, Inheritance, Variation and Evolution. And this tutorial will cover part 1, Reproduction, Genes and Gene Expression. We'll also be looking at mutations and meiosis. The rest of this topic can be found in the other tutorials. Starting off with DNA. DNA stands for deoxyribonucleic acid. And it is a unique genetic code found in the nucleus of our cells. Inside the nucleus we have chromosomes. If you unwind a chromosome you find the DNA. A section of DNA is called a gene, and each gene codes for a sequence of amino acids, and amino acids join together to make proteins. All of your genetic material is called the genome. So what does DNA look like? Well, DNA is a polymer and is made of two strands twisted together in a double helix shape as shown here. Within that structure, you have a sugar phosphate backbone and bases. The bases match up accordingly. So, A base always matches with T base, G base always matches with C base. These bases stand for cytosine, guanine, adenine, and thymine. DNA is made of nucleotides, so each nucleotide, shown here, is a sugar phosphate backbone and one base. The order of these bases is what makes a gene, and a gene then codes for a specific amino acids, which then join together to form proteins. Sometimes some parts of the DNA are switched off, so DNA controls what part of the gene is expressed or not. The order of the bases in a gene codes for the order of amino acids in a protein. Each amino acid is coded for by 3 bases, and that's called a triplet codon. Amino acids join together by folding into a unique shape, and then they make proteins. There are about 20 amino acids, but these join up together to make thousands of different proteins. And where this happens is in the ribosomes, and it's called protein synthesis. Different proteins include enzymes and hormones and various other chemicals in the body. So how does protein synthesis happen? Well, it happens in the ribosomes. Now, DNA needs to get this unique code to the ribosomes, but the problem is DNA can't leave the nucleus, it's too big. So instead, it copies its code to another molecule called messenger RNA or mRNA. When it copies that code, the DNA unzips and copies it across. That's called transcription. The messenger RNA then can leave the nucleus and travel to the ribosomes. When it gets to the ribosomes, another molecule called transfer RNA or tRNA then brings the correct amino acid that matches up to the code, to the order of those bases, and that's called translation. The amino acids then join up together to make different proteins. This whole process is called protein synthesis. Sometimes there's a random change in the DNA, and that's called a mutation, and this can be passed on. There's an increased chance of mutation if you're exposed to certain chemicals like carcinogens and radiation like UV radiation. A mutation can cause a change in the protein that's made because it can change the active site, for example, of an enzyme, so a substrate doesn't fit into it. There's 3 different types of mutation. Insertions, when a new base is inserted into the DNA, this is random. Deletions, where a base is randomly deleted. Or substitution, when one base is changed to a different one randomly. All of these can affect how the bases are read and then has a knock-on effect on the amino acids that they're coded for. So it can have a change, an effect on the protein that is finally made. As mentioned before, all of the entire genetic material is called the genome. Scientists carried out a 12-year project to determine all of the genetic material in humans, which is called the Human Genome Project. The good thing about this is it improves the diagnosis and research of genetic diseases. So different genetic diseases like cystic fibrosis, they can find out which gene that's on and research that more. It also allows us to research more into common ancestors and the discovery of the earliest ancestors in Africa. However, knowing what every gene your body codes for could lead to some concerns. For example, if you do know that you have a genetic disease or a carrier of a genetic disease, it could cause increased stress or to be affected by things like insurance, which is called genism, where somebody discriminates against you because of the genes. So here's some questions. If you pause the tutorial now for 10 minutes and attempt these questions. So what's the structure of DNA? Well, it's a double helix shape. It's got a sugar phosphate backbone and bases that pair up, and these are called A, T, G, and C. So A matches with T, G matches with C. It's a polymer with repeating units, and those units are called nucleotides. Each nucleotide is made of a sugar phosphate part and one base. What is a mutation? A mutation is a change in the DNA code. This can be an insertion of a new base, deletion of a base, or substitution of a base. And how does a mutation cause a change in a protein? Well, a change in the code of the DNA can cause a change in the amino acid sequence, which would then cause a change in the proteins that's formed from the amino acids. Moving on to reproduction. So sexual reproduction produces genetically different cells. So as you can see here, the egg and sperm join together to make 46 chromosomes in every cell of an embryo. So all of these cells in an embryo would be unique and different to the first ones that you started with. The sex cells here are called gametes. So 23 in an egg cell, 23 in a sperm, join together to make a new living thing. Asexual reproduction is when all the cells are identical. So let's look at that in a little bit more detail. Sexual reproduction involves two parents. The offspring, or the young that are produced, are unique and genetically different to the parents. It also causes variation, which means that there's differences in the offspring that are produced. And that means that they will all be able to adapt and evolve and survive differently. So it also leads to natural selection. And this natural selection means that we can also carry out selective breeding, where we choose desired characteristics to breed together. Asexual reproduction is one parent, so all the offspring are identical, they're clones. There's no variation, and because no mate is required, it is faster. And it can be used for cloning, for example, cloning identical plants. Some organisms can reproduce by both types of reproduction. So, for example, the protist that causes malaria in mosquitoes, that reproduces sexually in a mosquito but asexually in humans. Fungi can reproduce both ways, as can plants. So plants produce pollen and eggs, so can reproduce sexually, but also if you take a cutting of a plant or the runners that they produce or a bulb, that's asexual reproduction. So that's a genetical copy of the plant. So, what is meiosis? Well, in a previous tutorial, we looked at mitosis, and that's cell division to produce identical cells for growth and repair. Meiosis produces unique different cells, and it's how gametes are made. Remember, gametes are sex cells, so sperm and eggs. And gametes are all unique, and they all have half the number of chromosomes of body cells, so we call them haploid. Meiosis occurs in the ovaries to make eggs or in the testes to make sperm. Meiosis involves two cell divisions as shown here. So the chromosomes copy as happens in mitosis, and once those chromosomes duplicate, cells divide once and then once again. And as you can see, there is some exchange of genetic material, and all of the offspring are unique. So they're different to each other. They're also different to the parent cell, and they contain half of the information as the parent cell. Now, going back to reproduction, meiosis happens to form the egg and the sperm. Once the embryo is formed and starts to grow, that grows using mitosis, so the growth and development and copying of normal cells. And once it gets to a certain stage, the cells start to differentiate and specialise and form different types of cells. So a 6-mark question for this topic would be this one here. Explain how DNA codes for making proteins. So in other words, how are proteins made and how is DNA involved in that? Or what is protein synthesis? So if you pause the tutorial now and give yourself 10 minutes to answer this and think about the steps that we talked about in protein synthesis. Okay, let's go through some of the things you could include in your answer. So first of all, you need to mention that this happens in the ribosomes. Remember, DNA needs to get its code of bases to the ribosomes, but it can't leave the nucleus. So it copies the code by unzipping and copying it to a molecule called messenger RNA, and that's called transcription. The messenger RNA then leaves the nucleus and travels to the ribosome. Then another molecule called transfer RNA brings in the correct amino acid that matches up to the order of the code. So there's one amino acid for every 3 bases. This stage is called translation. The amino acids then fold up to make different proteins. There are about 20 amino acids that make thousands of different proteins. In summary, we've talked about DNA, protein synthesis, mutations, meiosis, and reproduction.